which is my journey, a bioinformatician journey to understand allergy. Okay, so of course, allergy.
course.
I want you to. Are you able to hear me, Ricardo?
Yes. Now we are watching your screen.
Okay, thanks. Sir. So I am specifically talking about a bioinformatics journey towards allergy because a disease can be studied in different perspective and different disciplines and different skills from the uh, uh, different from skills point of view. Okay, so the first thing is what is allergy? Allergy, like as we know, it is in many forms. It is many forms. It can be food allergy or smoke allergy or allergic to a tablet or allergic to anything chemicals, right? So and why I'm specifically interested in allergy, because 150 millions Europeans right now suffer or live with an allergic conditions. It could be either asthma, allergic rhinitis, or food allergy, or etc. So how do people currently identify allergy? One, with the allergic test, so which is called skin prick test. So they put a chemical on your skin and then to see if you have any allergic reaction to that chemical. And then other one is using blood to make an immunoglobulin E test, allergic test. So and the second one is what we are interested in, like for example, if a person is contacted with allergens or foreign substance chemicals, how does our immune system reacts and whether it produces immunoglobulin E or not. So by measuring this immunoglobulin E levels, we have an understanding of the allergic situation for any person. So a bit of basic before what is going on, how we are studying. I am studying this allergy from a human omics perspective, that is genomics or proteomics or transcriptomics. So the basic journey of a human phenomena, biophysiological phenomena. To put it in a perspective, our DNA, which is okay, our DNA, which is made of around 3.2 billion sets of genetic letters, which we have very four letters and sometimes five letters. In these 3.2 billion sets of genetic letters, each cell produces proteins that needed for it activities using the genetic information contained in these sets of genetic letters and produce proteins for each function. And these 3.2 billion sets of genetic letters produce across 20,000 genes spread across 23 pairs of chromosomes. And these 20,000 genes, and they transform into protein through mRNA, another process which transforms DNA into protein. So which produces around hundreds of thousands of different proteins, which is the process and the recipe required to our human existence and in living. For example, how we look or how we like the height, how the height differs between two persons, or how the skin color difference or hair color difference. These are all depends on the proteins, right? So this we measure for this is the genetic information which is collected through the blood, and the information collected from DNA is called genomics, and mRNA transcriptomics, and protein proteomics. We combine all these information and analyze to see what a person is having an allergy or not, how he is having allergy, what is the reason behind having this allergy, right? So when we combine these many letters, for example, when we see in our let's say in our computer, when we save some lines of letters, like if you write one essay of 10 lines, how much it happens, like how many letters, like 100 letters, if we say how many kbs, right? So in that sense, when we write all these 3.2 billion sets of letters in a text file, it will become a huge file, and which we cannot see from our naked eye. So we need some special programming and some clever techniques to understand what's going on. And also, how can we identify this, what is really causing a disease or the phenomena underlying allergy? Is it will be done in a perspective where we pull large number of data from many people. For example, like let's say I have a allergy condition, so I if I go to the doctor and he checks my situation using one of these two tests and understand it. But when it comes to research to set a guidelines to how to identify a person whether he has these allergy or not, we need to collect information from hundreds of people. Especially in the genomics research area, people collect samples, blood samples from thousands of people. For example, in our study where we had this genomics data, we collected information from 70,000 people who said they don't have allergy, any type of allergy, and also 50,000 of people where they said that they have some form of allergic condition. So we take blood from these peoples and then sequences, which means the procedure which results in the letters, like what kind of letters each person has across these 3.2 billion sets of genetic information. And then we put, we compare for all these like healthy people and sick people, what is the differences across these long length of genetic letters, where is there is a difference, correct? Once we see there is a difference between these people, then we perform certain statistical test, mathematic, we employ mathematics there and statistics there to understand what we see is really a, a cause of this allergic phenomena, or it is a fake alert we have got. Okay, so as a summary, we use this big data to analyze this big data. We use computer programming, statistical methodologies, and network analysis to identify a process what is causing this allergy, to understand a little bit more than what the current doctors or the researchers know. Okay, and which is absolutely not like this. So we won't use the computers like this to identify how analysis is coming, or it's not like plug and play kind of thing. So what I do most of the day, it is like this. I stare at these screens, black screens, multiple screens, and try to understand what is going on behind these. To, as I mentioned, there is these 20,000 genes and hundreds of thousands of proteins involved. So what it happens, like, is it a single protein is involved or single gene involved? So we take each gene into account. First we take one gene to understand whether it is causing an issue or not through mathematical and network analysis perspective, and then we see is it a protein, and then we found that none of a single protein or a single gene is causing allergy. So we thought it might be due to the combination of proteins involved in our human physiology. Like, it means like each protein talk to each other and then see what happens within that, if this interaction between and the talking between each protein is causing the allergic phenomena, correct? So, and why we should why this is important. For example, in our daily life we could see that some of the people having same characteristics do the same action. For example, let's say in our school, like we tend to sit, for example, when we enter the class after 5 minutes after the class started, and then we see there are like five empty places in the room, but we go and sit in a place where some of our friends or some of people who have similar thoughts are sitting. So we go and sit with them and spend rest of the day at the school. Even it applies to our daily life, or when we go to the pool or mall or movie theater, we tend to spend time with the people with like-minded characteristics, correct? Similarly, we
2 minutes,
employed
Pradeep,
this process.
please.
Yeah, so to identify a set of a processes, like for example we identified three processes where these is completely different. Like this is one is signaling between in certain proteins, and some involved in transportations from one cell to another cell component, and then other ones. So like this we identify three processes which is strongly involved in the allergic process. And then, as I mentioned, this is our bioinformatician perspective using computers, and we work with now our colleagues to see which of these processes is involved clearly, like which is first and which is second and which is the consequences of the disease. And thank you very much. If you want to have a more a deeper understanding of the work, you can scan this QR code. You have the whole research written in an article report format. Thank you very
Thank
much.